Symposium is Sam Slowinski, who will also be talking about parasites. And take it away, Sam.
Hello, thanks everyone for coming. As many of you are probably already aware, biparental sex is the dominant mode of reproduction across most plant and animal taxa. And biparental sex, it turns out, for those of you who just heard Mandy's talk will already know this, biparental sex is associated with a twofold cost. And given this twofold cost of biparental sex, we expect that self-fertilization, which is a uniparental form of reproduction, should be able to invade into outcrossing or biparentally reproducing populations. As depicted in this cartoon, in which a selfing mutant arises in an outcrossing population and rapidly spreads to fixation. And theoretical work suggests or predicts that this spread of selfing into outcrossing host populations and fixation of selfing can occur in as little as tens of generations. Yet, as I said before, biparental sex is so common in nature. So why is that? This leads me to my overarching question. What prevents the spread of self-fertilization into outcrossing populations? The red queen hypothesis is a leading hypothesis to explain the maintenance of biparental sex in nature. It posits that coevolutionary interactions with parasites impose selection against common host genotypes, which perhaps could favor the production of genetically variable, outcrossed host offspring. And my question is whether this selection for outcrossed offspring could be sufficient in natural populations to prevent self-fertilization from spreading into outcrossing host populations. In order to assess how, in order to study the evolution of breeding system, I used the model organism Caenorhabditis elegans, which is a great model for studying the evolution of breeding systems for two reasons. The first reason is that C. elegans hermaphrodites are capable of multiple modes of reproduction. Hermaphrodites can reproduce by self-fertilizing their own eggs, which is a uniparental form of reproduction, or by outcrossing with males to produce offspring. The second feature of this system that makes it a great model is that when hermaphrodites reproduce by self-fertilization, almost 100% of their offspring are hermaphrodites, whereas when hermaphrodites reproduce by outcrossing, they produce a 50-50 sex ratio of male to hermaphrodite offspring. And this relationship between the reproductive mode and sex ratio of the offspring allows us to accurately estimate the frequency of selfing and outcrossing in populations based on male frequencies in the populations which we can easily measure. And in fact, previous work in this study system has provided evidence that coevolutionary interactions with parasites can select for increases in the outcrossing rates in mixed mating host populations. However, the specific hypothesis that coevolutionary interactions with parasites can constrain the spread of self-fertilization into obligately outcrossing host populations is still largely untested. So to test this hypothesis, we designed an experiment in which we genetically manipulated the C. elegans breeding system to produce obligately outcrossing populations, and then we introduced mixed mating C. elegans or wild-type C. elegans capable of both self-fertilization and outcrossing into these obligately outcrossing populations at a starting frequency of 5% mixed maters as depicted in these pie charts. Then, having created these populations with multiple breeding systems, we allowed our experimental populations to evolve under different parasite treatments, and following experimental evolution, we measured the self-fertilization rate in our experimental populations so that we could assess how the presence of a parasite in the environment affects the spread of self-fertilization into outcrossing host populations. So just briefly to describe how we went about manipulating the C. elegans breeding system in this way, we had mixed mating populations. This is the wild type or natural form of C. elegans populations. And the mixed mating C. elegans express this wild type allele at the FOG2 locus. And I'll refer to this allele as the mixed mating allele because individuals with 2 X chromosomes that express the mixed mating allele will have a hermaphrodite phenotype, and individuals with 1 X chromosome will have a male phenotype. And then to create our obligate outcrossing populations, we used a FOG2 Q71 allele, which I'll refer to as the obligate outcrossing allele because it blocks the production of viable sperm and hermaphrodites, functionally transforming hermaphrodites, or XX individuals, into females by forcing them to outcross with males in order to reproduce. And I'll just point out a couple other features of this breeding system that we've created. One is that the male phenotype is unaffected by the genotype at the FOG2 locus. And the other feature is that hermaphrodites and females can both freely outcross with any males in the population, regardless of the male's genotype. And so these 2 different breeding systems that were competing in our experiments are able to freely intermix. In order to assess how parasites affect the evolution of host breeding systems, I used the parasite Serratia marcescens, which is a soil bacteria, and it's highly virulent against C. elegans. So all of our experimental treatments evolved on these Serratia selection plates. Before I describe the individual treatments, I'll walk you through our general experimental evolution protocol. So Serratia selection plates are agar plates that are seeded with the Serratia marcescens bacterial parasite on one side of the plate and with E. coli on the other side of the plate. And E. coli functions as an avirulent food source for the nematode hosts. In our experimental evolution protocol, the parental hosts were introduced onto the Serratia side of the plate where they were exposed to the pathogen. Those individuals that survived exposure could move across the plate to the E. coli food source and reproduce. And then after 4 days, we took a random sample of the individuals that survived and reproduced and used their offspring to transfer to the next generation Serratia selection plate. The only difference between our treatments, so all of our treatments evolved on Serratia selection plates, the only difference between our treatments was the source of the Serratia. In our control treatment, we used heat-killed Serratia populations as indicated by this fire symbol. So these heat-killed populations were dead, they're avirulent, they're unable to infect the host. And so hosts are being passaged in the absence of a virulent parasite. In our fixed parasite treatment, we took the same stock population of the parasite out of the freezer every generation, as depicted by this icicle symbol. And so in this treatment, the hosts are being passaged and they're able to evolve, but the parasites are not permitted to evolve. It's a static parasite environment for the host. And finally, in the co-passage treatment, both hosts and parasites were permitted to evolve and potentially to co-evolve. And in this experimental treatment, Serratia marcescens cells were extracted from the dead bodies of nematodes that died in response to parasite exposure, and then those extracted cells were used to seed the next generation Serratia selection plate. And so in this way, we were able to select for resistance in the host while simultaneously selecting for virulence and infectivity in the parasite. We ran 12 replicates per treatment, and we ran this experimental evolution for 33 host generations. In our control treatment, we predicted that given this twofold cost of sex, selfing should rapidly increase in our host populations. In our fixed treatment, we predicted that, that selection might initially favor outcrossing while host populations adapted to their novel parasite environment, but following adaptation to this static environment, the selfing rate should eventually increase. And finally, in our co-passage treatment, we predicted that the selfing rate should be constrained throughout the entire experiment due to coevolutionary interactions with this virulent parasite. I'll now show you our results in which I've plotted the host generation number on the x-axis and the host population selfing rate on the y-axis. Again, selfing rates were estimated based on male frequencies in the host populations. So because we manipulated our host populations to start with a 5% frequency of mixed maters, the selfing rate at generation 0 in all of our host populations was about 5%. In our control treatment, the selfing rapidly spread into our host populations. In our fixed treatment, selfing remained rare initially and then rapidly spread. In our co-passage parasite treatment, selfing remained rare throughout the entire experiment. These results matched all of our predictions and suggest that co-evolving parasites can constrain the spread of self-fertilization into outcrossing host populations. Next, we wanted to assess whether the frequency of the mixed mating allele, which again is the allele which permits selfing in hermaphrodites, correlated with the selfing rate across experimental endpoint populations. So to assess this, we extracted DNA from a random sample of individual nematodes from each of our experimental endpoint populations. We used a polymerase chain reaction with FOG2 specific primers to amplify the alleles at the FOG2 locus. And then we used gel electrophoresis to separate the mixed mating alleles from the obligate outcrossing alleles. And this allowed us to determine the FOG2 genotype of each individual nematode in each of our samples from our experimental populations, and then determining these genotypes allowed us to estimate the allele frequencies in our populations. So I'll show you our results of the allele frequency assays in this graph in which I've plotted the frequency of mixed mating genotypes on the x-axis. So these are genotypes that have at least one mixed mating allele. The mixed mating allele is dominant. And I've plotted that against the selfing rate on the y-axis. And as you can see, we found a strong, highly significant positive correlation between the frequency of mixed mating genotypes and the selfing rate across our experimental endpoint populations, which suggests that the spread of selfing was tightly linked to the spread of the mixed mating allele. Overall, our results support the red-cream hypothesis. With that, I'd like to thank everyone who made this project possible, and I'd be happy to take any questions. Yeah.
Any chance that the mixed mating allele may be exposed to some sort of drive to the male in population where the female is only the male protein? How might that affect the
Yeah, so that's a good question. So the question was whether there might be some kind of drive or maybe a meiotic drive, if I'm understanding you right, that could help explain the spread of the mixed mating allele. That's something that my advisor, Kurt Lively, has been very interested in, that we are interested in considering and pursuing in the lab. And I don't think we have data at this point, one way or another, to address that question. So we'd like to follow up on that in further projects. I would say that I should clarify that prior to the start of our experiment, we introgressed the mixed mating allele into the genetic backgrounds of all of our experimental populations, and then we divided each experimental population into the 3 treatments. So we had the same starting populations for all of our 3 treatments, and in our study, I don't think that the genetic background of our different breeding systems could have confounded our results.
Thanks, Sam.